Hello everyone, in this video we will discuss about the molecular identification process of bacteria using repetitive PCR-based DNA barcoding with the box A1R primer. DNA barcoding is a molecular approach that involves DNA sequence analysis using specific markers. These markers can be utilized for bacterial characterization, including differentiation among species, strains, and serotypes. So, before we start, we would like to introduce our team members, Faizatul Mufidah, Daniel Eka Fahrudin, Rio Ferdinand, and Safira Ramadhani Azahra. The first step in this practicum was bacterial DNA isolation. DNA isolation was performed using the Geneate Presto Mini gDNA bacteria kit with preparation for gram-negative bacteria. The process began with cell harvesting by centrifugation at 14 to 16,000 times G for 1 minute. After centrifugation, cells form a pellet at the bottom of the tube, and then remove the supernatant carefully. This step concentrates the cell by pelleting them at the bottom of the tube. Next is the suspended pellet. 180 microliter of GT buffer was added, and the pellet was resuspended using a vortex until homogeneous. Then proteinase K treatment. 20 microliters of proteinase K was added, and the sample was incubated at 60 degrees Celsius for 10 minutes, with inversion every 3 minutes. After sample preparation, the cell lysis step was carried out. Cell lysis breaks open the cell membrane to release cellular contents including DNA. After that, add GB buffer and mix. A total of 200 microliters of GB buffer was added to the sample and mixed by vortexing for 10 seconds. The tube was then incubated at 70 degrees Celsius for 10 minutes with inversion every 3 minutes. During this step, the elution buffer used in the vinyl stage was also preheated. The next step was DNA binding. After incubation, 200 microliter of absolute ethanol was added and mixed thoroughly. If precipitates formed, they were disrupted using a pipette. The mixture was transferred into a GD column placed in a 2 ml collection tubes and centrifuged at 14 to 16,000 times G for 2 minutes. After centrifugation, the collection tube was discarded and replaced with a new one. The following stage was washing. First, 400 microliters of W1 buffer was added to the GD column and centrifuged at 14 to 16,000 times G for 30 seconds. Then, 600 microliters of wasp buffer was added and centrifuged again under the same condition. The column was centrifuged for an additional 3 minutes to dry the column matrix. The final step of DNA isolation was DNA elution. The GD column was transferred into a 1.5 milliliters microcentrifuge tube, followed by the addition of 50 microliters preheated elution buffer and incubation for 3 minutes. Ensure the elution buffer is preheated before use to improve DNA recovery. Finally, centrifugation was performed to obtain the eluted DNA. Centrifugation was performed at 14 to 16,000 times G for 1 minute. Centrifugation collects the eluted DNA in the tube for downstream application. After DNA isolation, DNA quality and quantity were evaluated using a nanodrop spectrophotometer. The procedure began by turning on the instrument and selecting the sample type, including DNA, RNA, or protein. Here we choose DNA. The upper and lower measurement surfaces were cleaned using link-free tissue, followed by calibration using 1 microliter blank solution. After calibration, 1 microliter of DNA sample was added and measured. The instrument measures the absorbance of the DNA sample and displays the concentration and purity results. Following measurement, the surfaces were cleaned again before analyzing the next sample. The measurement results were saved and the spectra were printed for documentation. Nanodrop analysis results. Overall, most DNA samples showed good quality with values close to the ideal purity range, which is 1.8 to 2.0 for the 260-280 ratio and 2.0 to 2.2 for the 260-230 ratio. However, sample 7 showed a 260-280 ratio below 1.8, indicating possible protein contamination. In addition, samples 5, 12, and 13 exhibited low DNA concentration, which may potentially affect amplification efficiency. After determining DNA quality and quantity, PCR amplification was performed using the Prox A1R primer. The total PCR reaction volume was 25 microliters, consisting of 10.5 microliters and FW, 12.5 microliters PCR mixed bioline, 1 microliter box A1R primer, 1 microliter DNA template. The PCR program started with pre-denaturation at 94 degrees Celsius for 4 minutes. This was followed by 31 amplification cycles consisting of denaturation at 92 degrees Celsius for 1 minute, annealing at 50 degrees Celsius for 1 minute 30 seconds, and extension at 68 degrees Celsius for 8 minutes. PCR was run with a final extension step at 68 degrees Celsius for 10 minutes. The final stage of this practical work was agarose gel electrophoresis. A 2% agarose gel was prepared by dissolving 2 gram agarose in 100 milliliters of 1-time TBE buffer. The solution was heated using a microwave until boiling and cooled before adding 10 microliters gel stain. The solution was then poured into the cast and let it solidify at room temperature. After gel formation, the gel was placed into an electrophoresis chamber filled with TBE buffer until completely submerged. A total of 10 microliters repetitive PCR DNA amplicon was mixed with 2 microliters loading dye and loaded into the wells. Electrophoresis was carried out at 100 volt for approximately 60 minutes. After the process is complete, the gel was visualized using a UV transilluminator to observe DNA band patterns. That's all for this video. Thank you for watching.